10,000 years ago, a deadly virus arose in northeastern Africa. The virus 10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected. But it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairy maids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family, but when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
Okay, so I hope you've seen some background to pox viruses, and now we will talk about a few of these, and I hope by the end of this session you'll be able to describe how pox viruses and also parvoviruses look like, a bit on the pathogenesis, especially for monkeypox, which is the current issue, clinical features for pox and parvoviruses, and a bit on their treatment prevention strategies. The main viruses that I would like you to understand are smallpox, monkeypox, and parvovirus B19. There are many others, of course, in these families, but we focus on the most important. I see people talking about human experimentation in the chat, but we'll get to that. So pox viruses, what are these pox viruses? They are large and they are complex. This is, as far as human viral pathogens go, these are the largest and the most complex. They infect humans, and there are also others that infect other animals, and there are some that infect both humans and animals. They typically present with very prominent skin lesions. They're called pox. And the transmission for the most important ones, like smallpox and monkeypox, is through inhalation or contact with infected animals, humans, or fomites. The examples are smallpox, which is one infectious disease in humans that has been eradicated by 1980. We have monkeypox, which has risen to prominence over the last 1 year or so, actually maybe 6 months or so, but has been a problem at least since 1970s. But we also have molluscum contagiosum, which you are likely to see as a practitioner or even as an intern, or me as a student. Viral characteristics for pox viruses. These are large, complex virions. This 200 nanometer diameter is about 10 times, and they can be as long as 300, about 10 times the size of the average virus. It's also about half of the smallest bacteria. So it's a really large virus. The one that comes close to it is herpes viruses, but the rest are fairly small, maybe 20, 30 nanometers. It also has a distinct shape, which is brick or oval, depending on the species. They also have an envelope, and they are special because they also have a sort of double membrane, an inner membrane and an outer membrane. And for that reason, even when the envelope is destroyed, the naked, not really naked, but the mature virions that are an inner membrane are also infectious. So I wouldn't really call them naked, but they don't have the envelope, the outer envelope, they have the inner one. They have a linear, non-segmented, double-stranded DNA, which is also very large. 200 kilobases is also about 10 times or more the average virus. So they are very, fairly large. And this long genome codes for many proteins with different functions, the most important being immune modulation and also replication of the virus. They replicate in the cytoplasm. I would like to hear a specific reason why you think pox viruses, being DNA viruses, are able to replicate in the cytoplasm. Please place that in the chat. The reason why pox viruses are able to, it has to do with a specific enzyme. Which enzyme is this? As you post, I'll move on. We'll come back to it in the replication anyway. Taxonomy of the important pox viruses that I'd like you to think about is shown here. The entire family, Pox Viridae, has about 22 genera with numerous species. But the ones that have been isolated in humans, as you can see here, in 4 genera. The most important, I think, is orthopox virus. This is the one that contains variola or smallpox, monkeypox, which is the current pandemic, and camelpox, cowpox, and vaccinia, which is a close relation to variola and has been used as a vaccine for variola or for smallpox, and now for monkeypox. Molluscpox is the virus or genus that you're likely to encounter. This is molluscum contagiosum, which is usually common in immunosuppressed individuals, especially people with HIV and especially children, but can also infect, cause transient infection in others. And it's usually transient and benign, except in immunosuppression, it can be quite, it can sustain for a while. although it doesn't really cause severe disease. Most of the times it doesn't even have a fever. Yatapox is actually a combination of two types of viruses, Yaba and Tana. Yaba is in Nigeria, Tana in Kenya, and the virus that has been isolated in humans here is Tanapox, again causing a mild infection. There's also Parapox virus, and here this is mainly cattle, sheep, goats genus, but sometimes in animal handlers or people who deal with cattle may get infected, especially off virus or even pseudocowpox. So these are mainly cattle viruses, will not usually spread very well in humans, but they do cross over for people handling them on a day-to-day basis. I mean, handling cattle, sheep on a day-to-day basis. So really the ones that you guys are likely to encounter is orthopox and molassipox viruses. The structure is very distinct. As you can see here, if we start on the, so let's start on the right because it's simpler. You can see a core, which is really the capsid containing the double-stranded DNA. It's dumbbell shaped, and it's surrounded by this core membrane, or the core wall, as it's called on this side. And you have an inner, this bluish line is an inner membrane. This IMV means mature virion, and they tend to be released on lysis, and the, what is it called? The inner membrane is studded with tubules. These surface structures here are tubules, and the only difference that, and also carrying enzymes, and these two lateral bodies that also carry extra proteins. And the only difference here is that this mature virion can be coated with an extra envelope, and this happens in the Golgi, I mean in the cellular structures, to coat it or to cover it or to encase it in an envelope. And so you have this, it tends to be extracellular enveloped virus, and this is not necessary for infection. Both of these are infectious. If you remove the envelope of, say, hepatitis B or herpes, it will not be infectious, but this envelope is dispensable. So the other thing about this virus that is distinct besides the shape is these lateral bodies on the side. You can see them, that carry a lot of proteins, some of them immunomodulants, others whose function is not clear. And then also they carry enzymes, which allow them to initiate transcription and basically replication in the cell cytoplasm. So it's quite a distinct virus. You will not see another virus with double membrane, or with, maybe there are some with double capsid, but this kind of envelope is only this one that infects humans, and the lateral bodies, and also the dumbbell-shaped core, and which is actually visible on electron microscope, is distinct. The pox virus electron microscope looks like this for some of the viruses. There are some that will be more blocky, brick-shaped, but this is oval-shaped, and you can see the dumbbell-shaped core with a prominent membrane. The angle of the camera, of course, we may change the shape, because if this angle is across, it might show you something like this, depending on what level it was taken. So remember, the shape of the camera also matters. Not the shape, the angle of the camera also affects the shape. So the replication, I really broke this down so that I can highlight the most important bits. The first thing is that pox viruses replicate in the cytoplasm because they carry the DNA-dependent RNA polymerase. And for most viruses, other viruses that have DNA genomes will carry their own DNA polymerase, but they do not have the RNA polymerase that is required to transcribe the DNA into messenger RNA. And for that, they have to go into the cellular nucleus, which carries, or which has RNA polymerase that converts DNA into RNA. So remember, there is the RNA-dependent RNA polymerase in the cytoplasm, but that's not what you're interested in if you're a DNA virus. There is attachment and fusion as entry, but fusion can also occur through macropino or pinocytosis, which is a form of endocytosis. And after that, there will be uncoating, and then you have replication occurring in 3 phases. One is the early phase, where the promoters of viral replication are produced, and these are especially the proteins that suppress or modulate the immune system. After that, we'll have the replication enzymes as intermediate phase proteins. These include the DNA polymerase and the RNA polymerase. And so this will promote transcription and even replication. And in the late phase, then you have structural proteins being produced to go into assembly. And then there will be a little modification after that for some of the viruses in the cell before they exit through lysis. And this is usually late in the infection. Initially, there will be exocytosis and extrusion. Extrusion occurs through this cell transportation system called actin tail assembly. If you look at the diagram on your right, you can see the first row on top here. Move with me so that you don't get lost. The 2 viruses are different in slightly. One has a membrane or the envelope, which is what I was calling an enveloped virus. And the mature virion on the left doesn't have it, but both of them are infectious. And they can bind on the cell wall, on the cell membrane. The receptors are not very clear. So there are some proposed receptors, but I'll not go into that. But they use the tubules, the surface tubules to bind. And they can also go through endocytosis. After binding, they basically get internalized by the cell through endocytosis. And after that, there will be uncoating to expose the genome of the virus. So that is entry. We have dealt with it. There's a bit of uncoating here. And then once you have uncoated the virus, there will be transcription. And this occurs in phases. The early proteins, which include growth factors and immunomodulants. Then the intermediate proteins, the enzymes mainly, which are basically the replication complex. And then there will be late transcription of the proteins that make up the virus. Remember, the virus has many proteins, so these are quite a number. And they form the structure, the capsid, the protein, that is the protein that protects the nucleic material, the genome, and others in the lateral bodies, as well as the internal membrane of the virus. Now, as this occurs, initially, most of the viruses will go into the Golgi apparatus where they are wrapped as a membrane, wrapped using a membrane, and then they'll be exocytosed. So this is the initial stages most of the time. But over time, as the infection takes root and more viruses are being produced, lysis will occur. And then after lysis, these viruses that have not yet been wrapped will be released. But because they have the internal membrane, they are still infectious. And that's why they're called mature virions. The other mechanism that has been touted is this, which extrudes the viruses actively using actin tail assembly. The monkeypox is the virus that I would like us to focus on in this group. And this belongs to the genus, as the same genus as smallpox, which is orthopox virus. It is a virus that we've known for a long time, but we've ignored because it was first identified in monkeys, hence the name, in 1958. I think these were some monkeys that have been imported from Singapore, but the detection was done, I think, somewhere in Europe, Denmark, if I'm not wrong. So it has been known for a while. But despite the name, the natural host is thought to be rodents. It's not monkeys. Monkeys are an intermediate host. The first case was detected in humans in the DR Congo in 1970. And this was because at the time, there was a global drive to eradicate the smallpox. So they were mopping up any cases that look like smallpox, and as we'll see shortly, these lesions are very similar. So it was tested, but it was found to be nothing else. People were excited, no, it's not smallpox, so we can move on, but there was no effort to eradicate it. And it would have been challenging because given the natural reservoir, it would be difficult given the vastness of the tropical forests in Africa to eradicate monkeypox in the reservoirs. The virus is known for years to be, since 1970, to be endemic in Central and West Africa. And so far, until recently, it had just caused a few isolated cases outside Africa. But this year, there has been a pandemic that has caused thousands of cases globally. You can see here in this map, I liked it because although it's a bit old, July 2022, it shows us the area where the virus is endemic. And you can see here, these are countries that have a tropical rainforests. Initially was connected, but now some of it has been destroyed. This is DRC, you can see South Sudan, you can see Central African Republic, you can see Congo, Brazzaville, Gabon, Cameroon, Nigeria, all the way to Togo, Ghana, Cote d'Ivoire, and Liberia, and Sierra Leone. These are countries that have a significant amount of tropical rainforest where these reservoirs are thought to be. And there's also a test for game meat, different forms of game meat. So because of those interactions, they tend to be seeding and reseeding in the population, which occasionally spreads to other countries. You can see the spread in the latest pandemic outbreak has affected the West much more than Africa, actually, with the countries in Europe, especially in the Americas, taking quite a hit. I think so far there has been more than 70,000 cases, 77,000 cases, but the deaths have been few, 36 deaths so far. This is an article in the New England Journal of Medicine that describes the transmission cycle, so to speak. And if you look at the initial column here, you can see the circulation within the reservoir hosts in the rainforests. And then this could transmit to intermediate hosts who they interact with regularly, the monkeys. And these monkeys and even intermediate hosts could transmit it to humans, especially who are hunting. And usually during dressing, there's a lot of handling and some bit of injuries and bleeding. Dressing is basically slaughtering. If it's whatever animal you're slaughtered, it could be a monkey or whatever it is. And during that period, then the virus could jump over into humans, and then they will also expose their families now through direct contact. Usually once the food is cooked, of course, the virus will not infect those who are eating it, but those who are dressing it are at greatest risk. Dressing, slaughtering, and preparing it for sale or for eating. And this continues circulating. If it's a remote village in the DR Congo where it will take you 3 or 4 days or a week to get to Kinshasa, that may not really affect the rest of the world. And that has happened for a long time. Isolated outbreaks. Congo occasionally, DRC Congo occasionally has up to 1,000 cases in a year, but they never really get into this pandemic stage where after it skips into towns or into cities, because of the high number of people involved, transmission across humans is easy. If it's in a small village, not so much, but in the cities, we can see the explosion of the networks. Somebody with the infection, say in Lagos, Nigeria, travels to the UK, or they transmit it to somebody else who travels to Spain, causing an outbreak there, and all that leads to more cases in the US or Canada or Australia because of the connectivity, as compared to the village in Congo, which might be very remote. But there have also been cases, and you know, there is a lot of interest in shipping exotic pets to different parts of the world. And there were cases where pets shipped to the US infected prairie dogs, which then infected humans. So there is a bit of zoonotic virology here, which I hope is coming across. So by the time you're detecting the outbreak in humans, really it started a while back in the reservoir hosts and the peri-forest areas where people living next to the forests. So just now that we've understood the distribution of these viruses, it's easier now to understand how it has 2 different clades: the Central African or Congo Basin clade, which has been reported in DRC and South Sudan, who are neighbors, and it's more virulent with a higher mortality rate of about 10%. And then the West African clade, which has 95 to 99% sequence homology, depending on the isolates. Some of them are more different than the others. And it's been reported in Nigeria and other West African countries. I'm sure you've seen the distribution along the tropical rainforest. It is the current cause of the ongoing global pandemic. Mortality is fairly low. Overall, it's considered to have a mortality rate of less than 3.5%. Oh, sorry, I went backwards. The pathogenesis. So this is highly dependent on the site of infection. For the oropharyngeal route, and remember this could be through inhalation or through contact, coming into contact with people for intimate, especially intimate contact. It could be through kissing or through oral sex. And there is local replication in the epithelium, and this initial replication doesn't cause any symptoms, the initial one, in the mouth or in the mucosa. And there will be then local spread to lymph nodes, especially the inguinal lymph nodes, including the maxilla, submandibular, basically the cervical lymph nodes, and even the inner ones like the tonsils and what have you. And the initial replication causes low-grade viremia, which initially seems to spread lymphohematogenously into distant organs like the spleen and the liver, where there is amplification of the replication, causing a secondary major viremia. So the secondary viremia is major, it's not low grade, and it disseminates the virus to other organs and the rest of the body, such as the lungs, kidneys, and skin. And this then will cause damage through direct cell lysis and also immune modulation. Sometimes it can cause actually hyper immune reaction, but mostly it will suppress the immunity, and that can cause super infection, especially with bacteria. And this is actually what causes death. When people are, especially you can imagine the lungs are destroyed and you get a pneumonia, that can easily cause death. These are an important slide because I thought the immunosuppression or immunomodulation properties of monkeypox is important. If you are keen on immunology, you can read this paper, but I'm going to summarize it in these 4 points, as was done by the authors. This prevention of cellular signaling. Remember, cells are always signaling whether they are healthy or infected. And this is done through the MHC molecules that are expressed on the surface. If the cell is expressing MHC that shows it's infected, T cells attack it. If the MHC stops showing MHC, the natural killer cells attack it. But if the virus is able to dampen this signaling, it may be able to replicate in the cell without being attacked. And it does that through various mechanisms. You can see here blocking of pattern recognition receptors, which are usually useful for allowing the immune system to recognize a cell that is infected. You can see additional interference of apoptosis, which means that if the cell was going to die because it's infected, it is not, so it allows the virus to replicate, because if the cell dies, then the virus cannot replicate. It requires the cell. So if the cell is, instead of being triggered to die or to undergo apoptosis, remember apoptosis is the cell death without inflammation. So in this case, apoptosis will not occur, and so the virus will continue to replicate. There's also antagonism of immune mediators, and you can see here things like interferons. And interferons are the molecules that our cells produce when there's a virus infection, for example, which warns nearby cells to produce antiviral proteins. And there's a long signaling pathway, including this, you can see here, NF, kappa B, IRF3 signaling. All these promote interferon production. And so if they are blocked, then you have what we would call the antiviral effect is reduced. So if a cell was going to warn others to produce the antiviral proteins, then they are not produced, or pro-inflammatory proteins. And also the virus has been shown to have proteins that bind to things like tumor necrosis factor and other mediators of the immune system and it binds not to activate them, but to prevent them from activation, so that they do not recruit other cells to come there and, or they do not basically trigger the immune response as it's supposed to happen. So there's a lot that goes on in the immune system during infection of monkeypox. It's an interesting area of study, ongoing study. If you're interested, take a look. Clinical presentation is following infection occurs about 5 to 21 days, but it's usually a self-limiting illness for most people, with the common symptoms being fever, headache, and muscle pains. In the respiratory tract, some people may present with this also: sore throat, cough, shortness of breath, especially the lungs have been hit, lymphadenopathy or swelling and usually painful, especially of cervical or inguinal. So cervical if it's oropharyngeal transmission, inguinal if it's sexual or transmission is around the genital area. Rash, and this is usually local, but then it spreads across the body. And we'll see it's a particular rash, how it looks like. Pain and loss of function at the infection site. When I show you some of the images, I'll let you think through what could happen at those sites. And rash is usually progressive, starting with a macule. A macule is just a discoloration of the skin. It could be erythematous, or it could be this discoloration of any kind, which is flat. And then it becomes raised and fleshy, forming a papule. And then the papule will then become vesicle, meaning it now contains fluid. It's no longer fleshy. And then that fluid will become pus, forming a pustule. And then once the pustule bursts and forms a scab, then it will eventually reepithelialize and heal. So these usually are specific lesions, and you'll see them. Once you see them, I think you'll never forget them. Complications are also possible, and these include bacterial superinfection. Remember, we say that most deaths are due to superinfection. And you can also have in a few people encephalitis and septicemia. Septicemia mainly from the proteins of this virus that trigger, especially cytokine storm. And when pregnancy occurs, when infection occurs during pregnancies, you can also have miscarriage or stillbirth. This is a picture from New England Journal of Medicine, and you can see these particular pox, which are fleshy raised. They're in between, they haven't formed vesicles yet, but you can see the umbilication at the center, or the dimpling at the center, which is characteristic of pox viruses. When you learn about parasitology, we learn about myiasis, which are basically maggots in the skin or elsewhere. They also have a dimpling, but they tend to have a slightly different appearance, so don't confuse the two. But this is really very characteristic of pox viruses, be it smallpox or monkeypox or even other pox viruses, even molluscum, although molluscum tends to be smaller in size. The images here show you how different, depending on the stage, the lesions can look like. On this foot, plantar aspect of the foot or sole, you see a lesion that is on day 1 of the lesion, although it's day 10 of the illness, but you can see it's very difficult to tell apart or to tell what it is. You can actually ignore it. But if you look at the rest of the body in this human or in this person, you see on the forearm near the wrist, very distinct lesions that are umbilicated and even erythematous. And in the forearm, this is between the elbow and the wrist, you can see more lesions. One is clearly umbilicated, the other one is not. And then after, this is on day 10 still. On day 12, they are beginning to scab. And this is around day 15 of the illness, but day 12 of the lesion. So you can see if this patient is coming to you very early, they may just have a fever, maybe a rash that is not very distinct. So it's good to have a high index of suspicion and take a good history of the person. And this is a guy who was on social media to document the monkeypox infection. And you can see initially on, say, a few days after infection, by maybe day 1 of the lesions or day 2, they were fairly non-distinct or nondescript. And this is day 4 days following this, you can see they are now becoming a bit more prominent. 7 days later, You can see fairly distinct. This is 10 days later, they're beginning to form scabs. And about 11 days after that, also very similar. This is 13 days or about 2 weeks, now becoming quite mangled, very difficult to tell. They just look like scabs. By the 3rd week, there is not much. The person seems to be healing very well. So this is somebody who documented his story, and you can see it in this link. This is published in the Lancet in August, and you can see a lot of this was associated with men who have sex with men, and a lot of it was transmitted through contact. There has been documented virus in semen, but ejaculation is not necessary for this infection to occur. You need any contact of any kind can cause infection. And you can see this in the crotch, very distinct lesions. The ones on the tongue are not so distinct, but they are monkeypox viruses. This one may not be clear. Let me see if I can zoom in, but they're quite tiny, but also fleshy and painful. This guy on the trunk, this is panel D. You can see also not very distinct, but if you look at it there, you can see the umbilicated lesions, smaller than you expect. This is the anal region. These are the kind of guys who will suffer from tenesmus or inability to pass stool because of the pain, and a lot of them had to be decompressed because they couldn't pass. A lot of these guys would require a lot of analgesia to be able to pass stool, otherwise they suffer a lot of constipation and also infections. The lesion on the lip, this is a mixture of lymphadenopathy and also lesions at the preauricular area. And this is lesions, the white lesions here, you can see if you look at them, they have a central necrotic center on the back of the throat or the pharynx. This was in the glands and the shaft, on the skin of the penis. So very different, and this could be the initial presentation without the rest of the lesions. This is not one person, these are different people. So the presentation may be very different depending on the person and the route of transmission. This is a child, actually a neonate on day 10 of life with an uneventful birth. He was born in April 2022, somewhere in the UK. 9 days later, he developed this rash. On looking back, his dad, 9 days before his birth, had had a fever and a rash which had disappeared before he was born. The mom got a similar rash 4 days after he was born, and the child got this rash 9 days after birth. They survived after care. So how do we diagnose? Maybe I should pause here for some questions. Without looking at the charts, give me a moment to digest. I know there are many comments that I need to respond to. Human experimentation should be legal. It is legal if you follow ethical rules of research, but the risk has to be managed. You can't do some of the things that previously scientists did. So ethics becomes very important. A small price to pay for salvation, greater good. It's not small. So you just can't do anything anyway, but you can do some experiments. Like when you do a clinical trial, that's an experiment. In case I die, kids will enjoy the benefits. I don't know which benefits Mugambi is talking about. I don't know how much wealth he has that he's worried about. I have a suspicion it's not much at this point in life. Yeah, so there was an answer that for the virus to replicate, if it's DNA viruses to replicate in the cytoplasm, it needs a DNA-dependent RNA polymerase. So that is the question. Clades of a virus is the same as strains. You can say so, really, but sometimes it's not interchangeable, because you will find like within clade, the West African clade, there are 2 different types. So for now, we can say yes, but it's not always equivalent. But there are types or subtypes of a virus species. Self-limiting means this infection most times will go away with or without treatment. It may, of course, cause you some discomfort, but it will eventually disappear. in most cases, I mean, with a death rate of less than 3.5%, you can see most people recover. Stops on its own, I think it's a good answer. The body clears it actually. Is chickenpox? This is a very good clarification. Thank you, Kifaro. Is chickenpox part of the pox virus family? No, chickenpox is a part of the herpes viridae or herpes viruses, and I think it was covered when you covered herpes viruses. So chickenpox or varicella is part of the herpes viruses. It's distinct from other pox viruses. So it's a good distinction to make. Thanks for that, Kifaro. Strain tends to be used to refer to genetic change. An interesting answer here from Abeng. Varicella rashes have no dimpling. No, they don't. Chickenpox is varicella zoster herpes viridae. Thanks for answering that. I don't know what Mbivia Mutunga is referring to. I don't know what she or whatever is referring to. Myalgia is muscle ache. How is it transmitted in units? Just in this case, it was not vertical transmission, but it can happen. In this case, it was contact. Remember, this is a virus that is transmitted through contact. And in fact, for this child, the rash started from the hands and the feet. And these are the things that many of us tend to play with, apart from the cheeks in babies. So this is through contact. The baby was born healthy, but on day 9, they developed a rash. But you can also have the transmission occurring in utero. And I'll show you what happens. I think I mentioned that you can have stillbirth or miscarriage. If the mother has monkeypox while giving birth, will the child get it through vertical transmission? Yes, it could happen, depending on the viral load and where the infection is. Of course, if it's a single lesion somewhere on the arm, it may not necessarily get to the baby. The systemic immune activation, the cytokine storm, yes. Is it an STI? In a sense, it is now, but it's more than an STI. It can be transmitted without necessarily any sexual contact. So sexual transmission is just one of the routes, and it's not the major route. Okay, so how do you diagnose pox viruses? The specimens are numerous: throat swabs, skin, urine, and blood specimen. It's not clear right now which one is the most important, but depends on where the lesion is. If you can get the lesions, then you will have very good chance of getting the DNA. And the mainstay of diagnosis is PCR for nucleic acid detection. Sometimes you can do paired sera for IgG, which is detectable from day 8, and you can also do IgM detectable from day 5. But these are not, for example, if you do and you miss it, do not necessarily mean that the person is negative. Zinc smear, which is also used for herpes viruses, and it's basically staining with things like methylene blue or GEMSA, and looking for giant multinucleated cells on light microscopy. And if you see those, then you know you might be dealing with the pox virus or even herpes viruses. They can also, viral culture can also be used, but most of this is not for routine diagnosis. Immunohistochemistry is basically staining for the virus in tissue, and electron microscopy, which will show you the brick-shaped or oval-shaped virions. The management is that there's no specific antiviral treatment that has been licensed for monkeypox, although there are some that are in use that were shown to work for smallpox, but none has been licensed for monkeypox. The smallpox vaccines, because these guys belong to the same orthopox genus, they offer cross protection, and for other pox viruses, other orthopox viruses such as monkeypox, and there are two types. One is replication competent live vaccinia virus, and it's called ACAM 2000. But there's also attenuated modified vaccinia virus. There are two of them, MVA-BN, and these are just names. Modified vaccinia Ankara, BN is Bavaria Nordic, somewhere in Germany. And it's a vaccine that is non-replicating, meaning that it can be given to people who are immunosuppressed. The LC was developed in Japan years ago. This one is minimally replicating. So we encourage the latter the attenuated vaccines because they are safer. The live vaccinia virus vaccines have a bit of side effects, including myocarditis and others. So, and they may not be used in people with immune suppression. The LC, because has been around for a while, has actually been licensed in Japan for children, but MVA is only for adults, MVA-BN. You can see here the primary skin response or reaction after primary vaccination. And remember in this case, or remember I didn't mention this, the vaccination for monkeypox or smallpox is by use of a bifurcated needle. I don't know if I have it. Bifurcated needle without injection into the blood, but into the skin several times, 10 to 15 times, with that bifurcated needle. And the idea is to cause some scarring and elicit an immune response, deliver the vaccine, the virus there to replicate and then elicit an immune response. So that can cause this kind of response where there is local replication or local immune response causing damage to the cells, all the way from small pox to larger pox and then to a scarification or scab formation. And that is just from the vaccine. Prevention for monkeypox is vaccination for exposed and at-risk persons. This does not mean we give all of you vaccines like we did for COVID, because the risk is not as big as for COVID infection. Surveillance, contact tracing is very important, especially in this situation where the vaccine seems to be, not the vaccine, the infection seems to be within certain circles, social circles. And isolation of the sick, case finding also, for example, you find one, you can contact trace to find other cases and then manage them. And also post-exposure preventive vaccination, which can be given within 4 days, ideally, but can also be given up to 14 days if there are no symptoms. So these are some of the prevention strategies recommended by WHO. This ring vaccination is a strategy that I thought I should mention. It is what was used for smallpox, and even monkeypox now is encouraged to use it, because you can't vaccinate everybody. Once you identify a case, you identify the people who are exposed first to this person and give them the vaccine, then use them to identify the second exposure persons, which person they come into contact with. And so by doing that, you create a ring around the person who was infected, so that the infection does not get out of this ring to infect others. And if you find another one, you do the same. This is what was used to eradicate smallpox. And it is the same as been recommended. Actually, it's also been used for Ebola and is recommended for monkeypox. Okay, maybe a short break by looking at the chart before we move into variola or smallpox. Differences between antiviral and vaccines. Antivirals are drugs, vaccines are vaccines. So we have these as distinct topics anyway, and we will look at them when the time comes. The vaccine names, yes, I gave the vaccine names, but I'm not so sure you need to remember them. You just need to remember that some are replication competent and the other is not replication competent. ACAM 2000 is the replication competent one. MVA, BN, and LC16 are the attenuated ones. Okay. So smallpox is also called variola. Major and minor are the two types that were recorded, or were the two presentation forms. Variola major had 20% or more mortality. Variola minor had 2% mortality or fatality rate. In the 20th century alone, this is the 1900s, all the way to 2000, it killed 300 million people, which is a huge number of people. And we saw in the video earlier that Edward Jenner started variolation in the 18th century using cowpox, which was basically infecting people with cowpox, so that they could, which was known not to cause very severe disease, but was known to confer immunity against smallpox. So by infecting somebody with cowpox, you are essentially vaccinating them or immunizing them against smallpox. The global eradication program began in the 60s using ring vaccination, as I showed you earlier in the previous image or illustration. And by 1979, 1980, eradication had been achieved. There is still a concern for its use as a bioweapon. In fact, currently it is stored in 2 labs, I think 2 official labs. One is in Russia, Vector Lab, and another one is in the US. And this is because need to be prepared with vaccines and whatnot in case there is need or there's an outbreak. The clinical features are fever, malaise, centrifugally distributed rashes, basically beginning from the face downwards, and these are still following the same path: macules, papules, pustules, and scabs. And it could also affect the lungs and cause pneumonia-like infections. So a lot of deaths were occurring from that, but also the extensive lesions on the skin could cause serious problems. I'll just show you an image of that. This is the progression of smallpox from facial lesions becoming bigger and more dense. And as you can see, over time, these images are the same person on the day indicated. Over time, the lesions will come from the face to the trunk and become bigger and denser over time. This is an adult with smallpox. You can see this kind of skin, lots of issues can occur. And you can also have limited or variola minor just on parts of the body without causing a systemic disease. The therapeutics or the things we can use to directly target smallpox include these 3 drugs. Cidofovir and brincidofovir are DNA polymerase inhibitors, and brincidofovir is a prodrug for cidofovir, so it's essentially the same drug. Tecovirimat is mainly known to interfere with viral maturation, and you can also give vaccine immunoglobulin, or VGIV, which is known to, especially for prophylaxis after exposure, known to prevent development of the disease. These are the same drugs that people are now trying out for monkeypox, but there has not been clear evidence that they help. Although, again, because these 2 viruses belong to the same genus, there's a good reason to believe that they might help. Molluscum contagiosum is the disease you're likely to see. It's also a different genus from smallpox and monkeypox, and this is the Mollussivirus genus of Pox Viruses. It's a benign infection, self-limiting without much issue. There could be a single or multiple lesions, and usually transmitted through direct contact, including intimate sexual contact or just direct contact. And there are same features of umbilicated lesions. I don't know if there's a nice one here, but that central dimpling is seen very clearly. They could be anywhere on the body. Some can be quite severe in people who are immunosuppressed. In children, it will be on the face, trunk, and extremities. In adults who are not immunosuppressed, you may see it in the groin and genitalia over a period of time, and then they disappear. But also, I've seen an adult with very many lesions, molluscum contagiosum, but they had a very low CD4. They had HIV on treatment, but CD4 was, I think, less than 100 for a long time. The immune system refused to come back up after HIV treatment was initiated. Parvoviruses, maybe a break again, as I look at the chart. This is what does VGIV do? It's an immunoglobulin. So immunoglobulin, if you give it after exposure, it will clear the virus and prevent infection. It's the same, similar to rabies immunoglobulin or hepatitis B immunoglobulin. Septicemia due to immune response of viral toxins. The viral proteins trigger septicemia, so they trigger an immune response. And so the 2 are working together, because cytokines, once triggered, will then cause circulatory collapse. The toxins don't work alone. They work by triggering the immune system to cause septicemia. Is there a visual difference in clinical? No. You can't tell. You can't tell the difference between monkeypox and smallpox. The only reason you will say it is not smallpox is because you know smallpox has been eradicated. But it could be any other pox virus. If somebody, for example, is an animal handler and they present with pox lesions on their hands, you may be dealing with off virus or pseudocowpox or cowpox or monkeypox. So you can't tell until you test with the PCR. So the PCR may be targeting the genus orthopox virus and with a specific gene for monkeypox. So if the monkeypox gene is negative, but the orthopox genus gene is positive, then you know it's a different orthopox. It could be cowpox or any other in that genus. So keep that in mind. Parvoviruses. Parvo means small, and difference between bacteremia and septicemia. Bacteremia is just bacteria in blood. It's like viremia. Septicemia includes circulatory collapse. Parvoviruses is parvus or parv means small, and these are quite small. Remember, we said for pox viruses, the nanometer diameter was about 10 times this. The size of parvovirus DNA is 6 kb or 6,000 base pairs. It's a single-stranded DNA as compared to pox, which is double-stranded. For pox virus, it was 200 base pairs. So you can see quite small. They are naked or non-enveloped, and there are 5 genera out of 26 that infect humans, and only 2 are confirmed to be pathogenic. They are widespread worldwide. In fact, there's a study we did in 2016 in Taita Taveta samples, and we found close to 80%, 70 to 80% antibodies, meaning exposure was quite high. The genera that infect humans are listed here. And remember, not all these are pathogenic. Boca parvoviruses, which includes human bocavirus 1 to 4, are thought to cause pneumonia or have been isolated in the respiratory tract. Erythroparvovirus is the one you need to remember. Erythroparvovirus includes parvovirus B19, which can actually cause death in a few people. And these are in this, the official name is primary erythroparvovirus 1 and 2. There are also protoparvoviruses, and this include buffavirus. This one, even if you forget, I will not blame you, but it's just to show you these are more recent viruses in this group that have been discovered. I think buffavirus was found in stool of children with gastrointestinal, with diarrhea basically. Whether it was the cause or not, we are not sure. Tetraparvovirus includes parvovirus 4, which is also a recent discovery. And all this you can see, they go by the name primate and the corresponding genus name. So primate erythroparvovirus, primate protoparvovirus, and primate tetraparvovirus. Now, an interesting group is the last one, the dependoparvoviruses. They are called dependoparvoviruses because they do not replicate on their own. They require adenoviruses to replicate. And these are actually used as vector for vaccines in research. So right now there are vaccines that are being developed using these dependoviruses as vectors because they do not, so they are modified because they do not pose any danger to humans. They are not pathogenic. I just put them here to show you that there is more to parvoviruses or not, than infections. Actually, one of the things we do now as we do plasma virome analysis, when you're looking at which viruses have you found in the plasma of humans, parvoviruses are so many, and sometimes they're actually coming from the kits that you're using to do the analysis. So having a parvovirus in the body is not necessarily pathogenic. The other thing we saw is in cancer cells, which are highly replicating, there's a likelihood of parvoviruses there. And they are not really causing the cancer, but they're there because these are fast replicating cells and they provide a good place for the virus to make copies. So they are an interesting group of viruses. So the one that I would like us to focus on is parvovirus B19, which is the one that is likely to have a clinical presentation that you want to know about. B19, as we said before, like any other parvovirus, is single-stranded DNA, which is naked. It infects red blood cell precursors. by using this receptor, which is called the P blood antigen, also globoside. And the transmission is primarily through the respiratory secretions, could be saliva or any other respiratory secretions, and infection will lead to a lifelong immunity. Mostly, the illness is self-limiting, but can also cause death. The fancy name for this is erythema infectiosum. Some people call it the fifth disease, and it can be asymptomatic in a good number of people. This is the simple structure of a parvovirus. You can see the linear single-stranded DNA surrounded by a capsid, which has two proteins. And these are VP2, which is this blue structure here, and VP1. As you can see, it's called a minor capsid protein, and VP2 is major capsid protein. But really, those are the two structural proteins. The important gene that this genome codes for is the two structural proteins, VP1 and VP2, as well as the DNA polymerase, which is coded by a gene called NS1. Compare that with the complex structure of the pox virus. Here, this might look very complex, but it's really very simple. If you start with me at the top here, you have a virion or parvovirus, which attaches to the globoside or the P antigen. It is enters the cell through endocytosis as an endosome here, and then it will enter it will uncoat and enter the nucleus. In the nucleus, there will be transcription and replication. You can see the different genes being produced, and there seems to be a role of hypoxia, which seems to upregulate the transcription of these viruses. That is what this thing means. So hypoxia seems to initiate some signaling that then upgrades or upregulates the transcription of parvoviruses. And you can see here the DNA polymerase making copies of the genome, and then the genome is transcribed into messenger RNA. This messenger RNA will code for different proteins, mainly NS1, which is the enzyme, and PVP1 and VP2, which are capsid proteins. And what happens is that these messenger RNAs will go into the cytoplasm. They'll be translated into proteins. The proteins will be imported back into the nucleus, and they will be used in assembly. So by the time the virion is leaving the nucleus, it's already assembled. The genome is already protected by the capsid, and then it will cause lysis and leave the cell through lysis. So really, you have entry through endocytosis, replication in the cytoplasm in the nucleus, and then assembly within the nucleus, and exit through lysis. Does smallpox cause lymphadenopathy? Yes, it does, but not as much as distinct as the monkeypox. That seems to be quite a distinct feature. But we do not have smallpox anyway, so you're not going to see it now, unfortunately. Fortunately for you. The main difference between smallpox, yeah, that is true, but it is considered a big difference because monkeypox is more likely to cause lymphadenopathy. Thank you, Harun, for answering that. The next thing on parvoviruses that I'd like to look at is incubation, not incubation, presentation. The incubation is usually about 1 week. A macular rash appears on the cheeks and may present as slapped cheek syndrome. I'll show you this and you'll remember it because it's very difficult to describe. Diffuse maculopapular rash may also follow the local or the facial rash. It's not always present, but it can happen. And it may also present as glove and sock syndrome, which is a reddish rash on the hands and the feet, which is clearly demarcated at the wrists and at the ankles. And you can also have arthralgia, although arthralgia, sometimes with arthritis, is more frequent in women, adults, more than children. This here is a baby with the malar rash or the slapped cheek syndrome, but also generalized or diffuse rash. This one is very distinct malar rash. This one here is the hand glove syndrome, very erythematous rash in the fingers and in the hand. You can also have this, you can see a malar rash without the generalized rash in the rest of the body. Presentation in special categories, aplastic anemia, and this is what you should be aware of when you see parvovirus infection, because this is where your doctor will come in. Aplastic anemia may occur in patients with hemoglobinopathies. In our case, this is the sickle cell disease, and you may cause, or the virus may cause transient aplastic crisis. This can actually cause death if not intervention is not done. Hydrops fetalis may occur if infection occurs in utero, and chronic anemia may occur in immunosuppressed persons who do not clear the virus. So if the virus is in people with cancer or people on immunosuppressives or immunosuppressant agents, then they may end up with chronic anemia. So there are also some cases of myocarditis, vasculitis, glomerulonephritis, and encephalitis, which are quite rare, but they could occur. Lab diagnosis is mainly through PCR. Serology for IgG, which could be paired, may also be used, but IgM has been a challenge because there is no standardized assay for it, and this could be through ELISA or IFA. There is also no specific treatment, so treatment is supporting. So for example, if somebody has a plastic anemia, then you manage as a plastic anemia, which means they might require some transfusions and other, if they have any other conditions causing it. There is also no vaccine for parvoviruses, mainly because it may not be commercially viable. There's not much motivation to use it. There are several sources that I've put here, there are others that I have not, but I thought these ones may be useful. So you have this case scenario I'd like you to post in the chat. You have been called to attend to a 27-year-old male with fleshy umbilicated lesions on his forearm. Just post in the chat what action you can take, what actions you're going to take. This is very open. I just want to see what your thoughts are. How does hypoxia come into play? As I said, patients with, say, for example, hemoglobinopathies will have hypoxia, and the hypoxia triggers some cell signaling that seems to promote DNA expression or DNA transcription of this virus. If you are familiar with the JAK-STAT pathway, then you can take a look at it. It's one of the triggers of the JAK-STAT pathway, but you can also look at specifically how hypoxia promotes expression or replication of this virus. Is B19 infectious? DNA, no, unless it is with the capsid. Do chronic hypoxic conditions present in disease scenarios make you prone to this pathogenic replication? Yes, because the virus will replicate, and if the person has hemoglobinopathies, because the virus attacks RBCs, then they'll be exacerbated. If the person is immunosuppressed, then they will have chronic infection. Isolate, disinfect the area, collect sample for testing. I don't know which sample you'd like to collect, but isolation is good, disinfect is good. What else? That's a good start. Supportive care, take to ICU. Oh man, the guy has 2 or 3 lesions, why do you want him to go to ICU? But avoid direct contact as much as possible. So how do you avoid direct contact? It means use PPE. Use personal protective equipment, gloves, a gown, goggles, and even a mask. Isolate and do so you can get a swab of the lesion and do a PCR test. You can see Edna and Kidenji, Sindel have proposed that. Christopher, what is the mechanism behind Hydrops fetalis? It's because this virus infects the erythrocytic cells or erythroblasts. And actually in the fetus, it also infects the heart because the globocyte receptors are also in the heart. And in this case, you will have anemia, which then causes problems with the liver and other organs, and eventually will cause hydrops. This is mainly because the virus will attack and lyse cells that are in the erythroblastic stage, but also because this child is developing, they have a lot of these receptors in the heart as well, the fetal heart. Is it recommended? They can see a contact tracing. Very important, Zakaria Abdi. Talk to this person, find out which circle have they been in, who have they come into contact with, and can these people come in and get tested? So that's a very good thought, Zakaria. Salo, skin specimen, do PCR. You can just swab this text and do a PCR, comprehensive medical history. Is it recommended to poke the lesions? You don't need to poke it, you just swab it, there will be a lot of DNA on it. Is the case is dash monkeypox, seal. Yeah, for now, I would like you to believe it's monkeypox because that's what is circulating, but you can't tell unless you test. Can monkeypox present with warts? No, but there have been cases of warts being super infected with monkeypox because warts are in the genital area, and if this person gets infected with the monkeypox in the genital area, they can still get infected. So there have been cases. There have actually been a few publications on that. Okay, so if you have any other questions, I'll still take a look at them. I think I can stop the presentation there in case we have a few more questions. I can take this for another 5 or 10 minutes. Yes, hydroxyphosphate is the same as seen in alpha thalassemia. So this is when the baby has very severe anemia, and so the organs are affected. So you can't differentiate visually between species of pox viridis. No, you cannot. How can you tell the difference between chickenpox and monkeypox? Chickenpox does not have dimples. It is not umbilicated. It's just vesicular lesions. Although it can be macules or papules, so at that point it may be difficult. But the best way is to test and also get the history from the patient. Umbilicated lesions from molluscum contagiosum, yes, molluscum contagiosum was umbilicated lesions, so they don't. They are very similar, but they're smaller than other pox viruses. And you're likely to see molluscum very easily. Just keep looking. How will the macular rash look like on dark skin? It will just be, I don't know if I have an image for that. Let me see. It could just be discoloration, but also reddening or darkening of the skin. This is something here that looks close to it. If you imagine this on a dark skin, or of course on the soil, which is lighter, it will appear, but you can see the macules will suddenly either be darker or lighter or red on the skin, depending on the the tone of the dark skin. I don't know if there is another lesion here on the dark skin. You can see here on this top right corner, although these are now beginning to be raised, but you can see there's a bit of a difference on the skin. There are a few other images I didn't put here, but if you check online, you may see distinct lesions. I think I didn't put some here, but you will be able to see the markings even from other illnesses like measles and whatnot. They do not, they're discoloring. The color is different from the rest of the skin. I'm looking for the chart now. I think I lost it. Thank you. Somebody has asked a question. Why do adenoviruses used? Why do adenoparvoviruses use adenovirus? They cannot replicate on their own. So this must be an evolution event where these viruses were able then to patch onto adenoviruses and begin to replicate. We see it with hepatitis D for Delta virus, which requires hepatitis B to replicate. Is pure red cell aplasia associated with B19? Well, I think aplasia can occur with B19. I'm not so sure if pure red cell aplasia, I've not checked what pure red cell aplasia is caused by, so I can't comment on that. But B19 can cause anaplastic crisis or chronic aplastic anemia. I'm not sure what pure red cell aplasia is caused by. You can check that with the hematologist, or you can check it for yourself. Yvonne, if you want to attack me, do so directly. Kifaru, why do you have a notice streak? What actually is the immune complex deposition? What actually is the immune complex deposition in hand and knee joints associated with leukemia infectiosa? It is usually the such viruses will trigger antibodies that tend to destroy other proteins in these places, either in immune mimicry, like we see with a few other viruses, and sometimes just deposition itself can trigger efflux or coming influx of cells and other immune modulators into the area, causing inflammation. And remember, when inflammation occurs, for example, a neutrophil and whatnot, they destroy everything around there, not just the virus. They have a limited genome and thus need virus encoded replication to replicate. That's part of the answer, I guess. How do we do culture monkeypox and tissues used for culturing? We do not culture this in just a standard lab because of the risk of infection. So we require a bit of security, so P3 enhanced or P3 labs. But you can get any tissue and just culture it in cell lines, human cell lines. What is the most appropriate specimen for diagnosis? Right now, there's none. It depends on the presentation. So if somebody has lesions, that would be the best. But if the lesions are not yet there, throat swab would work because the initial replication is the oropharynx. You can also sometimes find it in blood, and antibodies can also be checked in blood, but that's not reliable. It has also been found in urine. So it's a mix of all this. So when you find a patient you suspect, you can pick all the samples. I mean the macular rash specifically for leukemia infectiosa, the one on the cheeks. Or for darker skin, it will just probably be darkening that area or reddening if they are light enough. And the rest of the body might have a rash that may not be as clear as the rest of the light skin, but you can still see it. That's why you require natural light. Mustafa, adenovirus donates early E1 protein. Now we are splitting hairs. Yvonne, did you mean baby face? I think I will ignore that. There's somebody who is really interested in Yvonne here. Maybe they need to have a sit down. So for the one with lesions on the forearm, what would be the most appropriate? For that, you just swab the lesion, but you can also get blood. But if you can swab that lesion, then you'll be getting a reliable sample. But sometimes they may come with the scars and you want to confirm the diagnosis. So urine would work very well. Okay. I assume there are no more questions. Thank you for listening. I see parvoviruses or pox viruses viremic. Yes, there is an element of viremia in both. And especially for pox viruses, there is a major viremia. Primary and secondary viremia. Okay. I see no more questions, so I'll stop there. Thank you for your time, and I hope you've learned something today. If you have more questions, I think we are still meeting. Let me see when our next class is. I think it will be on sequencing, if I'm not wrong, on Friday. So we'll see you then. Enjoy the rest of your day.
Thank you.
Asanteni pia.